Well, good morning, everyone, and thank you so much for the nice introduction. So we have got a good background about the whey and the whey permeate and all sorts of the whey components that we are dealing with right now in Wisconsin. So taking a step further on that, so I'm going to discuss about my research today that's on exploring the prebiotic potential of WPPC ultrafiltrate retentate in promoting the growth and its binding to a very beneficial bacteria in our human gut, as we all know, that is Limosilactobacillus reuteri. So to start with, we know, I don't need to give a very in-detailed introduction of how WPPC is obtained, but just going through, so basically we know it's a co-product of the whey protein isolate, and when we basically churn the cheese, separate it into whey, and then we ultrafiltrate and we go for the permeate and the retentate, and finally we get the whey protein phospholipid concentrate. And at present, we can, like the present US alone, production of WPPC is around 77 million pounds. So that's a huge number. Now, most of that is, as we also, got in the very first plenary section, we understood that most of that is now given as an animal feed, mostly taken by China as well for the animal feeding systems. And also, some of them are also used as ingredients for processed foods. So now, if it is already established as an animal feed, it is already given in the ingredients as in the processed food, why are we interested in WPPC? Now, there is, apart from the triglyceride core, that is the main structure of the WPPC, we also have the milk fat globular membrane, and to attach it is the membrane-bound glycosylated proteins. So that's the interest of my research here, that why the glycosylated proteins that are basically attached in the form of N-glycan and O-glycan form to the WPPC is something which have taken our interest into this research, apart from the glycolipids that are also attached to the membranes. So the hypothesis of our research came from the idea of the two previous works that are done by our group. The very first one was basically on the this one. The first one was done by in contest of the bifidobacterium using the WPPC as a growth medium to grow in the in vitro conditions. So basically, when WPPC was given, bifidobacterium was able to grow without any addition of any other carbohydrate or nitrogen sources. So that gave us an idea that WPPC may act as a growth promoting factor to the lactobacillus that are persisting in our gut. The second hypothesis was based on this study where we established that the most important bacteria of our gut, which is now known as the L. reuteri. So that organism had a very specific component to join to have the adherence capacity to the WPPC, to the intestine mucosal wall in our gut in the human beings. So based on these two observations, our hypothesis was that can we use WPPC as an adhesion to the L. reuteri and then make it available to our gut so that if it is acting as a carbohydrate or nitrogen source in some way, it can also be a protectant to the L. reuteri, which otherwise, when passes through the digestive system in the stomach, may get decreased in their numbers. So going forward with that, the first step was obviously to characterize the WPPC UF retentant. So after getting the WPPC, we again filter that, and after the ultrafiltration, we take the retentant section, and then we characterize that, and we can see that in the retentant, most are the glycosylated proteins of the xanthine oxidase and the glycem-1, which are the most of the glycoproteins that were present in our retentant section. And if you can see the green highlighted one, there is basically no lactose, no glucose, just a very minimal quantity of galactose, that is 0.2%, and 2.05% of the proteins. And we anticipated that most of these proteins are the glycosylated proteins, as I mentioned, seen in our SDS page. So moving forward to further characterize that by the LCMS, we could see that for the quantification, most of the xanthine dehydroxygenase and the glycem-1 were the most predominant proteins, glycoproteins that were present in our protein section. So further to see that, as I mentioned, that we wanted to see that in the bifidobacterium part, we saw that the N-glycosylated proteins were able to help that to grow. Now, in this case, we were also interested to see that whether the O-linked glycosylated proteins are able to help our L. reuteri to grow or not. And if we can see that the modified MRS, as we all know, that doesn't contain any glucose or any lactose in the sugars in that. So when we added WPPC, at the end of 8 hours, there was a significant difference in the growth when WPPC ultrafiltrate retentate was given as a supplement. Next, we saw that whether it can act as a nitrogen source and a fatty acid source. So we took the LDM3, LDM3A. So LDM3A means that is without any amino acids, and LDM3F means without any fatty acids. So you can clearly see that when LDM3A was supplemented with WPPC UFR, there was definitely some growth, which was again significantly different at the 8 hours as compared to just LDM3A. But fatty acid, in case of fatty acid, we could see that there was no difference in the fatty acid, even if it is supplemented with the WPPC. So that clearly demonstrated that WPPC UFR may act as a source of carbohydrate and amino acid source, but not as a fatty acid source for the L. reuteri. So going forward, we also did a gram staining to see its adherence properties. So basically, we just washed the LR cells, the wild-type LR cells, with or without WPPC, and then incubated it with WPPC for 1 hour at 22 degrees centigrade, and then harvested the cells. So you can see that if it is just WPPC, it's like just a cluster without any cells here. This is just the wild-type LR, just the normal cells. And when we add WPPC with the wild-type LR, there is a full cluster, which basically shows its adherence capacity with the WPPC. Now, the next part was that, okay, it is binding, great. Is it binding to the proteins? So to further see that, we did a SDS-PAGE, and we just compared the wild-type WPPC UFR with just the WPPC UFR. And we could see that there was a slight reduction in basically the glycan part of the proteins, which were the glycosylated proteins. And also there was a slight reduction in the concentration of the other glycan proteins, but this was not very clear that whether it is only adhering to the glycoproteins or the other proteins as well. So to further see that, we did a glycosylated protein specific staining for that, and from here we could very clearly see that so the glycostaining is basically staining, so whenever we have the glycoproteins, it will come in the pink color. Now, we can see that most of the glycoproteins here were taken up by the cell, because this was the cell-free supernatant which we started to run here. So the cell-free supernatant was basically the part which was free of the glycosylated proteins. And to further see the temperature variability of that, so we again saw that at 4 degree and 37 degree. And at 4 degree, we could see that there was a difference till the 8 days, the cells could pertain with the WPPC, but if we see a cell reduction, rate of reduction, the rate of reduction was not significant. So with or without WPPC at 4 degree was there was not a significant difference. But when we come to 37 degree, we could see that there was not only a significant difference in the persistence of the with the WPPC, for the wild type LR strain, but the rate of reduction was also very significant. So in this case, it was very evident that at higher temperatures, WPPC is certainly acting as a protectant somewhere to protect the wild type LR. So to just intensify with our study for the pH one, so we just did an InfoGest model to see that how it sustains the pH. And from there, we saw that so in the pH conditions, we know the stomach is having a pH of 2.8, whereas the intestine is like 6.8. So in the 2.8 condition, we could see that when the WPPC is supplemented, so the initial cell count here was 1 into 10 to the power 7. And you could see that there was a 1 log reduction in case of WPPC supplementation, while there was around 2 log reduction in case of only wild type strain. But when we took this sample and directly gave it to the SIF condition, which is 6.8, you could see there is like a huge log reduction in case of just wild type as compared to the wild type WPPC. So that means the pH conditions were sustainably maintained when WPPC was supplemented with the wild type strain. So to further validate that in the in vivo model, so this is a very preliminary model study that we have done now. And we could see, so this was like for 1 to 3 days, we treated the mouse models with just LR and just the LR with WPPC. And then after 3 days, we stopped the oral garbage and just collected the fecal samples from day 4 to 9 and just saw that what is the CFU count when in case the WPPC is given as a supplement or not given as a supplement. And we could clearly see that for the first 3 days when the treatment was going on, obviously there was no significant difference in the survival rate. But as the treatment was stopped, starting from the 4th day, starting from the 5th day, actually, the rate of reduction in just wild type LR was very significantly, like very high as compared to the persistence, so in case of the WPPC supplementation. So here we can clearly see the significance in the reduction rate, and the wild type LR reduced in a very drastical rate as compared to the supplementation with the WPPC. So all these results basically gave us some quick conclusions that were the growth support was given, definitely was like WPPC UFR definitely provided a growth support in case of like carbohydrates and amino acids. The binding mechanism was very evident with the glycosylated proteins, and in the stability enhancement, we could see that for high temperatures and low temperatures, the stability was still maintained more towards the higher temperature. Survival in simulated fluids, that is in the simulated conditions, was quite good. And in the in vivo persistence study, we could also see that the persistence of wild type with WPPC UFR was much higher as compared to just the wild type LR strain. So for future directions, we plan to see for the long-term storage stability studies and also for the mechanistic studies for adhesion, that is what mechanism basically directs the wild type LR to bind with the WPPC. So what is the genetic mechanism of that? So that's what we want to study further. And for the acknowledgements, I want to acknowledge Professor Gulistan, who is the chief PI of this project, Professor Susan Pan and Professor JP, who are my PI and co-PI respectively, Dr. Jiwan, who is also a very major person working in this project, and Dr. Zarina and Michael Armstrong, who are also the part of this project. So yeah, thank you. Yeah, I can see you there. Thank you.
Thank you. I think we have time for one question. Does anybody have a burning question? Does this sound like it's on? Yeah. Question. Yeah.
Thank you for the talk. That was very interesting. I think you demonstrated really nicely that WPPC is improving L. reuteri survival and providing some sort of beneficial nutrition. I'm curious about this in the context of the human gut, where there are, thousands of different bacterial species. Can you comment at all on what L. reuteri might be doing in the context of the human gut, or how this might benefit other bacteria?
Yeah, so in context of human gut, we already have studies which have demonstrated that L. reuteri has the adhesion capacity to the mucus protein in the gut already. So that we know now. But the problem is the transportation from the stomach till the intestine. So what we are trying to see is that if WPPC has an adherence capacity to the L. reuteri, so one, that may help, because it is a protein that will not be degraded in the stomach environment. And second, if it is also having the, the nutritive effect, so by the binding, it can also have the reuteri to go to the intestine, bind somewhere, and then also take the nutritional advantage of that. So that's what we want to see.